Hello everyone, welcome to today's live broadcast, Immunotherapy of Cancer using Genetically Enhanced T cells. I am Christina Mahalik of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational webinar presented by LabRoots. We have a few important announcements before we begin. This webcast is designed to be interactive, so we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can also enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing the presentation, please click on the support button at the top right of your presentation window or submit the problem through the green Q&A button, once again located in the lower left corner. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of the webpage and follow the process of obtaining your credits. I would now like to introduce today's speaker, Dr. Michael Milone, Assistant Professor of Pathology and Laboratory Medicine at the Hospital of the University of Pennsylvania, Associate Director, Toxicology Laboratory, University of Pennsylvania, Perelman School of Medicine. Dr. Milone received his MD and PhD in experimental pathology in 1999 from Rutgers, New Jersey Medical School, formerly known as the University of Medicine of Dentistry of New Jersey. After an internship in internal medicine at the Hospital of the University of Pennsylvania, he completed postgraduate medical training in clinical pathology, transfusion medicine, and laboratory toxicology. Following clinical training, Dr. Milone pursued postdoctoral research, training in cancer immunology and adoptive immunotherapy with Dr. Carl June at the University of Pennsylvania, where he performed basic research to develop CTL-019, a genetically modified T-cell therapy for B-cell leukemia and lymphoma that has been licensed by Novartis and undergoing early phase clinical testing. He is currently an assistant professor of pathology and laboratory medicine at the University of Pennsylvania School of Medicine. At present, Dr. Milone's laboratory is focused on T-cell biology and the development of new techniques for culturing and genetically modifying T lymphocytes for adoptive immunotherapy of cancer. I will now turn it over to Dr. Milone for his presentation. Doctor?
So thank you, Christina. Hopefully everyone hear me. So before I begin, I just need to get disclosure information. So as I think shown on the initial disclosure slide, I do have some intellectual property right been licensed to Novartis and have received royalties. Also, my laboratory receives funding for research from Novartis, as well as from others as shown on this slide, and all of these conflicts are managed in accordance with the University of Pennsylvania policy and oversight. So to begin, I think I want to stress the point that the idea of using the immune system to control cancer is really not a new idea. It's an idea that has been around for over a century. With some of the early work from scientists like William Foley, who's a surgeon in New York City, who made the observation in the 1800s, early 1900s, that some patients with cancer, they were infected with a particular bacterial infection, exhibited spontaneous regression of tumors, and he spent many years trying to sort of recapitulate that as a therapeutic approach. mostly successfully. However, the approach, did to probably the idea of what many people may recognize, which is vesicular BCMA therapy for a transitional cell carcinoma, similar kind of idea of using bacterial toxins to stimulate an inflammatory response for an anti-cancer benefit. The idea that cancer may actually be, regulated, and that we might actually prevent, cancer on a regular basis through immunity, is also not an idea. That's an idea that was worth, as you can see here, in probably over 50 years ago, by for example, Burnett, who hypothesized that when individuals that are, developing spontaneous mutations and changes in their somatic cells that could lead to malignancy, that there should be some mechanism to prevent that. And I think we know, certainly that there are, mechanisms to control that at a DNA level. In fact, since we're the week of Nobel Prizes, last year's Nobel Prize, was medicines focused on that. But he put out the hypothesis that maybe even immunological mechanisms might be at play to do this. And I think over many years, people tried to demonstrate this, with lots of conflicting data. But over the last, decade, really, I think we have become to understand that immunotherapy can, in fact, have benefits in cancer. So when you think about the immune system, the first question you have to think about is the immune system target? So in most cases, we're tolerant to self, and so you need something that you can differentiate tumor from a host. And as Burnett had hypothesized that maybe some of the genetic changes that occur when these tumors might provide them of that. I think the past, maybe 10 years, with next-gen sequencing, we've really begun to really understand the landscape of genetic mutation in cancer. And as shown on a slide, you can see that cancers really run a wide range of, the numbers of somatic mutations within their genome. With cancer like melanoma has the highest numbers of mutations per megabase, for example, of the genome. Same thing for lung cancer. Whereas other tumors, for example, if you look left, neuroblastoma and acute, leukemias have a much lower tumor burden, mutational burden. And so really their range, and even within some of the high tumor burden, as high mutational diseases like melanoma, there's a wide range with some having over hundreds of mutations, or others having, a couple that might be targetable. So we know cancers can, in fact, be naturally immunogenic, and that this has formed the basis of cancer vaccines targeting some of these changes. And in addition to those genetic mutations, there's been a lot of effort to try to look at tumor-specific, tumor-specific antigens such as the Endrian vaccine, Sipuleucel-T, or the GP120 vaccine, tumor-associated antigens, but not necessarily specific to the tumor. But I think what really shows us the benefit of potential tumor immunotherapy is that immune checkpoint inhibitors, which have gotten a lot of attention recently, melanoma and non-small cell cancer, really shown dramatic effects on cancers like melanoma and non-small cell lung cancer. But I think an important thing to recognize is that, again, the nature of those antigens and what you target may be critical to the success of those therapies. And in these two studies in the past 2 years that have focused on looking at the relationship between mutational burdens and the outcomes of checkpoint blockade, it becomes very apparent that the checkpoint blockades really require high mutational burden and a source of antigen to really provide that response. And so, it raises the question about whether the low antigen, low mutational burden diseases could actually be successfully targeted with therapies. That this is one of several barriers, and not all tumors, as we even have high mutational burdens. Looks like we're having some audio issues. So I guess I'm ready. If you're ready, I can switch the. I'm hopefully audio. Is there?
Hold on 1 second. We're dealing with some technical issues. Hopefully this is better now for the audio. And I think we should be good to go. All right, so going on to the next slide here. So in addition to that mutational burden that may not have the mutations necessary to target the cancers, I mean, there's many other barriers that we face with cancer immunotherapy. So we know tolerance mechanisms, which is one of the checkpoint molecules. For example, CTLA-4 probably targets the regulatory T cells. But there are many other checkpoints and modulators that, such as myeloid suppressor cells, that inhibit the inflammation within tumors and block tumor immunity. And then, of course, when we do cancer chemotherapy on patients, we induce a lot of immunosuppression from the nature of the drugs that we give patients. And then, of course, tumors acquire many local properties that further help them evade immunity, such as alterations in their ability to present tumor antigens. So even if there are mutations within those tumors, they can't present, as well as disruption of normal immune cell migration and activation through expression and modulation of chemokines, and creation of other barriers, for example, physical barriers such as the stromal elements that are within tumors. And so when we think about this, what my research lab and our group here at Penn, and I think several other groups around the country and the world, really have begun to think about how do we get beyond the limitations of the natural immune system, which clearly, if cancer has succeeded, and if it really is being held in check normally in most cases by the immune system, and when you get cancer, clearly that would indicate a failure of the immune system, that we might need something beyond the natural. And to use an analogy that I think is helpful is thinking about performance-enhancing drugs in sports. And so if you take this analogy of Barry Bonds, baseball player from Oakland, you can see, in this graph here of his slugging average, illustrating how, over the years, with his improved, training, reached some plateau in his slugging average, but really improved beyond what he was naturally able to achieve through the use of, basically performance-enhancing drugs. And so you can think of it as a bit of putting the immune system on steroids. We really need to find ways to get beyond what is naturally capable. And this is where I think the chimeric antigen receptor technology that I'm going to mostly talk about really provides one of the methods to do that. And so to really take what is a very complicated system and digest this down into hopefully something that is at least relatively simple, you can think of the T-cell receptor complex, which is how we naturally recognize antigens, as this very complicated receptor, but comprised of two components. One is the T-cell receptor, that recognizes antigen, and in the case of the T-cell receptor, it makes its recognition through the MHC, the major histocompatibility complex, and peptides derived from cellular proteins that are presented on the MHC, and then delivers its signal through a variety of molecules, but one that is probably sufficient is the CD3 zeta chain. So if the other chains are not functional, you can still get a functional T-cell receptor with just the CD3 zeta chain. And But we know that the T cells, in order to generate a really robust immune response, it requires additional signals beyond that, so co-stimulatory receptor signals like CD28. And these have to be, necessary to really get the cytokine secretion, the proliferation and survival of those critical T cell clones, and to mediate the killing of target cells. What we've tried to do with a chimeric antigen receptor is essentially take that complex T cell receptor complex and co-stimulatory receptor and really simplify it into a single, molecule that can be expressed within a T cell that would have the antigen recognition capability as well as the signaling. And this is done all in a single molecule, which actually amazes me every day, that this enormously complex T cell receptor and co-stimulatory system can be recapitulated at some level by this synthetic molecule. But nonetheless, it works. And through the use of monoclonal antibody technology, where we can now essentially make antibodies to proteins largely within a week, perhaps even less, through approaches like phage display. We could take essentially in the antibody molecule the two, what are depicted in this diagram as the purplish variable domains, the variable heavy and the variable light. You can combine them into a single molecule called a single chain variable fragment that we can then link up to this chimeric receptor, the CAR. And now we have the capability of really conferring the specificity of T cells under the control of a defined antibody single chain FV. And this now allows us to, one, generate very rapidly CARs to almost any target that we would want to target them to. Of course, the CAR approach does have a fundamental difference from the T cell receptor, which we can also clone from, if we know we have tumor-specific TCRs, we could theoretically clone those T cell receptors, and many individuals are doing this. But the difference is that the CAR actually allows us to target beyond just simple proteins. We can target non-protein antigens, for example, gangliosides. So there's been CARs made against the GD2 ganglioside that's expressed in neuroblastoma, as well as more recently, one of the groups here at Penn generated a CAR targeting a mutation that in cosmic that leads to an abnormal glycosylation of MUC1. And so these are kinds of changes that the T cell receptor could not do. The one limitation, though, is that the T cell receptor requires surface expression. The CAR requires that the target be on the surface of the targets of the tumor cell, whereas the T cell receptor can, in fact, pretty much sample the entire proteome of the tumor. So they are different but complementary approaches. And so how do we implement this type of artificial receptor in a adoptive T cell immunotherapy? So I'm going to basically show in this next slide what the general approach is to using these therapies. So we start with a patient who we can collect T cells from using procedures very analogous to dialysis, known as apheresis, where we can collect on the order of billions of lymphocytes from an individual. We can do gene transfer into these cells, and what we use at the University of Pennsylvania are lentiviral vectors, which are HIV-derived gene therapy vectors. However, people have done this with retroviral vectors, and there's also a lot of work looking at ways to use non-vector-based gene transfer, such as electroporation of RNA into T cells, transposons, and even now work or efforts to use gene editing approaches to introduce CARs into T cells. We can then take those cells, and if we have 1 billion lymphocytes, we can expand those ex vivo to greater than 10 to the 11 cells using ex vivo expansion, and then reinfuse. And to put that into context for folks who are not really immunologists, 10 to the 11 cells is approximately what you have in your body right now. And we can take that whole entire number of T cells and redirect their specificity towards a single target antigen, giving us a tremendous amount of cytotoxic potential. So this next slide really it serves to just make a fundamental point for those who are used to thinking about traditional drugs. And I think the CAR T cell is very different from most drugs that you could think of in that it's a living drug. And so what's shown in this image is basically a CAR T cell targeting CD19 And the green cells represent tumor cells that are expressing CD19, and that CD19, in fact, is tagged with a GFP marker. And you can see how the T cell clusters that CD19 at the interface where the CAR is interacting with that cell, and then very soon after that interaction, the T cell delivers the death signal, and that cell starts to undergo apoptotic cell death. And so what's important is that these are living cells that have the ability to replicate and therefore give pharmacokinetics, I put that in quotations, that it may be very different from any other type of pharmacokinetics you might think about for a traditional drug, be that a biologic or a small molecule. So as we were developing this, we were thinking about what would be the ideal target for a CAR. And I think, the list is, of sort of 3 characteristics that we think are probably going to be necessary to use this technology successfully and safely. Is number 1, as I mentioned earlier, the target has to be expressed on the cell surface. Number 2, the target has to be expressed on all tumor cells, because we can expect that if it's not on all tumor cells, you'll get selection. And number 3, it needs to be absent from normal tissues. And the reality is, as far as I am aware, there's not a single target antigen that we're currently pursuing that meets all of these criteria. But at the time, I think our group and many other groups realized that probably the best sort of, starting point for this kind of technology that really, at least meets 2 of the 3 criteria, at least we thought, is CD19. And so CD19 is a molecule that is restricted to the B cell lineage. So it's not absent from normal tissues, but it is, pretty much restricted to the B cell alone and no other cell type within the body. It comes up very early in B cell development and then spans the entire range of differentiation of B cells, even to the terminal plasma cell stage, where it does get downregulated, and so you have populations of plasma cells that are positive and negative for CD19. But of course, all malignancies that arise from B cells also retain this expression, more or less, providing us now with an antigen that we can theoretically, and it's on the surface of the cell, that we can target B cell leukemias and B cell lymphomas. And while it's not absent from normal tissues, we would expect it to target the normal B cells. The other thing that we knew is that normal B cells, if individuals have genetic defects, for example, in congenital Bruton's gamma globulinemia, where they don't have normal B cells, people can live relatively normal lives with IVIG immunoglobulin replacement, making the toxicity, at least we thought, manageable. So just want to point out, as I mentioned earlier, T cells require multiple signals. A signal 1, which delivers that sort of necessary signal to activate the T cell and cause the degranulation. And it also is necessary for proliferation, but it's not sufficient. So it's necessary but not sufficient for full T cell proliferation, which is what we think is necessary to get large numbers of these cells. So I think we knew that we need co-stimulation. Unfortunately, as shown on this right-hand panel, is that there is a very large range of different molecules that can deliver those proliferative signals. And on the next slide, you can see that as we've gone over generations of CARs, that really, it's been a largely empiric approach, but comparing different CARs with different signaling domains, that there are many ways that one could make a CAR. We chose to focus very early on looking at the CD28, which we know is a very critical co-stimulatory signal, and then the 4-1BB co-stimulatory signal, which is part of the TNF superfamily of receptors that is necessary for survival, and we know memory in CD8 T cells. But then there are also 3rd generations combining these multiple receptors, these multiple co-stimulatory domains as well. I don't have time in this presentation to really go into why we chose a 4-1BB domain that's in these published papers. But both CD28 and 4-1BB 2nd generation CARs for CD19 are actually both being developed by many different groups and companies, and I think which one is the superior one, I think, is still waits to be answered. But as I mentioned, we put forward 4-1BB largely because of what we demonstrated in a preclinical setting. It improved persistence of T cells in a leukemic model. So we initiated these trials, and the first patient we treated with this was a patient with CLL And we did that, I think, in part because CLL is a in a subgroup of patients can be very aggressive, is very highly CD19 positive, and in general, we also thought about acute lymphoblastic leukemia, but bone marrow transplant is used quite frequently there, and we felt in the early phase, to start at least, CLL was a better disease to begin to do this. We completed the first 14 patients, which was reported in last year. And you can see from this slide, and I apologize, unfortunately, it seems the color got changed to white. Hopefully everybody can see that. But basically we treated a population of patients who were relatively high risk in their disease characteristics. And in this cohort of patients, see an overall response rate of about 57%. With a complete response rate of 28% and a partial response rate of 28%, so put that together to get your 57, with a almost 40% of patients non-responsive. And I think what we learned from this study is shown on the next slide, is that really, as we suspected from the preclinical data, that long-term persistence and expression really is an important characteristic for this therapy. And so shown here is in a series of plots that are looking at the number of genetic sort of vector copies per T cell. So this is a marker basically of our engineered T cells in the blood over time. So time is on the x-axis. And this is a log scale, and you can see that very clearly, I think it's apparent that the first row represents our complete responders. Those are the 4 patients who showed a complete response. And then the patients who are partial responders, and then you have the patients who are non-responders on the bottom. And it's pretty obvious that the non-responders show very poor overall engraftment. And the area under those curves are much different from the area under the curve for the complete responders and the partial responders. And so what I think this is telling us is really that you do need that persistence and that engraftment of these cells to mediate the effect. And so it would be analogous to a small molecule where if you're not giving the drug and the drug is not really there with sufficient concentrations, you don't get the effect. We've moved on to other indications, and what I'm going to show you in the next slide is our work with, and I just want to point out that these cells can persist for a very long time. And so this is just showing one of our patients with CLL who was out over several years, where we were able to show that, number one, the T cells are persistent in those patients out to that time point. So even out to over 1,000 days, we still have detectable CD8 positive CAR T cells in the peripheral blood. And more importantly, when we take those cells and we actually test them against T cells, this is a degranulation assay and an assay for stimulation of MIP-1 beta as a cytokine secretion, a marker of T cell activation, that you can see if you let those cells interact with a negative target cell, you don't really see much in the way of upregulation of MIP-1 beta, nor do you see degranulation. But when you take them and mix them with CD19 positive target cells, they degranulate, which is a marker of their cytotoxic function, and they are very abundantly producing MIP-1 beta, showing that those cells that are present are still functional in the blood out to several years. We've moved this on into additional indications, and probably our most advanced disease indication that we have focused on mostly here at Penn is in the acute lymphoblastic leukemia setting. And in fact, we've done most of that in pediatric ALL. So this is showing data from the first 59 relapse refractory pediatric ALL patients, where we're seeing a CR rate of about 93% with a median follow-up at this time point that I can show you of 12 months. The important things to recognize here is that we really haven't seen many relapses, no relapses at this time point, past 1 year. However, we do have patients who obviously do relapse, and I will come back to that. But I want to point out that the response rates that we see with this CAR are comparable, at least, to other CARs out there. For example, the JCAR 017 that's reported out of Seattle is showing you know, a similar type of greater than 90% CR rate, as well as the a little bit lower in the ALL setting for the Kite CD19. However, nevertheless, we definitely see pretty good activity across different CARs in this disease setting. And I think the efficacy really in showing the persistence is illustrated. This is our sort of poster child for this therapy, Emma Whitehead, who really was the first child to receive this therapy, and she was fairly advanced in her disease, very short life expectancy, and I could say that now she's over 4 years in no detectable cancer with CAR T cells still persistent and present. Showing the real persistence of these cells. So again, the efficacy, though, doesn't come without toxicity. So I think to highlight and sort of summarize the toxicity that has been observed in these patients, B-cell aplasia, as we expected, is certainly a toxicity. It's observed in all responding patients to date, and currently is managed with replacement therapy, basically IVIG. Tumor lysis syndrome is another, not unexpected when patients have high tumor burden. However, I think the thing to point out here is that patients can have this complication, and it can be delayed. So we have seen it anywhere from 20, even out as far as up to 50 days post T-cell infusion, which is probably a little bit wider range than you might see with typical, small molecule chemotherapeutic drugs. Probably the biggest, most notable, I think most talked about toxicity is the cytokine release syndrome. And this is a reversible toxicity that occurs from on-target activation of the T cells. When they engage CD19 positive target cells, they produce lots of cytokines. And we know that the severity of this syndrome is related to the tumor burden. And what's important, I think, is that it can be quite severe. So it's something that really I don't think we expected until we treated patients, to the degree that it can occur. And in many respects, as we mentioned, a lot of our patients are pediatric ALL patients, and our pediatric hematologist oncologist, Stephen Grupp at CHOP, Children's Hospital of Philadelphia, has really likened it to the macrophage activation syndrome. When you look at these patients' bone marrows, you can see erythrophagocytosis, and it really parallels a lot of the features with incredibly high ferritin levels. But what's important is that it can be reversed. It appears with cytokine blockade, in particular IL-6. And so, our current strategies use tocilizumab to treat these patients. And Emma Whitehead, who was that first patient, really showed a dramatic response that was reported in that paper. And the other big thing, I think, to consider that we don't understand and didn't expect was the neurotoxicity, which in these patients ranges from delirium during the fever through frank encephalopathy with aphasia, confusion, hallucinations. It doesn't appear to correlate with the CRS severity and doesn't appear to be prevented by tocilizumab. But it does appear to be self-limiting, and so when it resolves, the patients usually seem to recover fully from this therapy. So just go into a little bit more about the cytokine release syndrome, because I think it is an important, and one of the challenges to really broadly applying this therapy. This is actually the data from Emma Whitehead. This is the CHOP 100. This is our first patient treated with this. And you can see that after receiving the CARTs on day 0, that within about 3 days she began having intermittent fevers to fairly high levels that in an adult would be quite severe. And then a few days after the spiking fevers, you start to see the LDH rise, indicative of probably tumor lysis. And if you and when she started spiking these fevers and she was getting some degree of respiratory and hemodynamic instability, we tried steroids, we tried blocking TNF using Enbrel, and unfortunately, I think because of pediatric indication of juvenile arthritis, tocilizumab was actually on the formulary at Children's Hospital. and that was tried really as a last-ditch effort. And it's quite miraculously they achieved from the way Steve Grupp describes it, within hours of her receiving tocilizumab, her fever resolved, blood pressure and most of her hemodynamic parameters improved dramatically. And basically, you can see here that much of that clinical syndrome really is associated with the spikes in cytokine release. And we've definitely seen that this pattern is very similar in CLL and in ALL. And I just point out and point to your attention that there has been a grading system that has been developed for CRS, as well as recommendations on when really to treat with interventions such as tocilizumab. I think we still, it's still early days with this therapy, and I think we, are still exploring the range of when you can apply the tocilizumab, and I am hoping that there will be trials to really look at even maybe prophylactic use of tocilizumab to prevent cytokine release syndrome. Since patients who get it during the peak of response still seem to have good responses. So, as I mentioned, this is just showing the disease burden, and it is well correlated. The grade 4 high, severe CRS is correlated with the level of disease burden. And more recently, I think in efforts looking back at the cytokine data, trying to look at whether there might be markers that might predict patients who are going to go on to have very severe CRS. And I think this is published data. You can see here from our experience in the ALL setting, you can see that we certainly can define a few cytokine markers that can be applied in the first 3 days of therapy that have a fairly high sensitivity and specificity for severe CRS. Unfortunately, we currently lack clinically validated methods to measure all of these cytokines. And the other big challenge is, even within that 3-day period, these patients often can progress to a very severe CRS fairly rapidly. And so whether you could get the rapid clinical turnaround necessary to use it is unclear. And I think that's where I think prophylactic or early intervention studies really need to be done. The other thing I need to discuss is really the mechanisms of resistance. So we know, as I mentioned, that we can get greater than 90% of patients into a complete response in both pediatric and adult ALL. However, 15 of our first 50 patients treated with ALL went on to relapse. And we know that 10 of these 15 patients relapsed with CD19 negative leukemia. And so this was, I think I talked about this earlier, that CD19, we thought it was going to be a fairly good marker, definitely for CLL and for mature B-cell malignancies. They pretty much uniformly express it. We did have this concern with ALL, that ALL patients can have, it can vary. Not all ALLs actually express CD19. And so it may be a result of the, the fact that CD19 comes on around the time, when we think the ALL sort of malignant cells arise from that state of B-cell maturation. Nonetheless, we know that we had 10 relapses, and so I think the question is, what are the mechanisms of CD19 negative relapse in ALL? And so in exploring that, we have seen that there are a couple of different ways. Surprisingly, most of these ALLs seem to still maintain CD19 expression. However, it seems the dominant ways of acquiring this resistance really are through two mechanisms. One is a splicing mechanism where we know that the epitope that we're targeting by the CAR, which is derived from the FMC63 CD19 clone, targets exon 2 predominantly of CD19. And so there's at least a couple of cases of splicing that occurred that removes basically that exon 2, presumably disrupting, and we know the epitope, and we know that cells, when we introduce this splice variant into cells, they're not sensitive to CD19, to the CTL019 CAR product. Mutations have also been identified in both the exon 2 and exon 4, and we believe that these may be hotspots that may be necessary for the antibody binding to these domains. So Many different trials have now looked at this. This is my attempt at summarizing most of those trials here. But you can see that really CD19 is being used quite widely with different signaling domains. So as I mentioned, the two dominant signaling domains are the CD28 and the 4-1BB. However, I think there are also trials now looking at even other types of signaling domains in this setting. So I guess I want to move on and look at how can we expand this technology beyond CD19. So certainly I think it works quite robustly in CD19 malignancies, but can we expand this to other target antigens? So one of the antigens that we're pursuing is the molecule BCMA, or B-cell maturation antigen. This is a molecule that is expressed on mature B cells, pretty much memory, a subset of memory mature B cells, but is very highly expressed by plasma cells. And it's actually thought to be important for maintaining plasma cell homeostasis. And much like CD19, this molecule, BCMA, doesn't seem to have expression anywhere outside of the B cell lineage. So as a logical extension to CD19, we think this may be one that will have a lot of similarities to the CD19 molecule. And so this first in human clinical trial was actually performed by Jim Kokendorfer and folks at the NCI, which was reported this past summer with the reference below in blood. And they showed that they could take 12 patients and they treated them with a range of cell doses. And really at the highest cell doses is where they saw their effects, and they had one stringent CR lasting 12 weeks as well as 2 VGPRs. So we have also been developing this target, have a clinical trial open at Penn. And one of the things that we're looking at in this trial, which I think differentiates us a little bit, is the question of whether we need lymphodepleting chemotherapy. And so we have in this trial built in 3 cohorts of patients. A first cohort that is actually receiving these CAR-T cells without lymphodepleting chemotherapy, which is somewhat, contentious, I think, in the field, and most, I think, would argue, and actually the neurotoxicity associated with the CD19 CAR that was reported and caused the halt in the Juno trial was, thought to be at least associated, perhaps, with the fludarabine lymphodepleting chemotherapy. We thought it's important to ask the question, do we need this lymphodepleting chemotherapy? So we decided to build this into our clinical trial, along with then 2 cohorts that received the Cytoxin. So we didn't use fludarabine in this setting because we were concerned about the toxicity in the myeloma patients who are often lymphodepleted already. And then this is an error here I just want to point out on cohort 2. We're looking at a lower dose of cells and then a higher dose similar to what we're using in cohort 1. So this trial has been open and we've been recruiting. The design, as I mentioned, is very similar to how we do most of our CAR trials, where we do an apheresis, collect the cells, manufacture. Patients get the Cytoxin day -3 if they're in the cohort that's going to get that. And then are receiving, and we've also decided in most of our CAR trials to actually do a split dosing strategy where we give 10, 30, and 60%, with the idea that if we give 10 and 30% and then patients on day 2 start to develop fever, we don't really want to fuel the fire by putting more CAR cells into that patient. So we felt that a split dose might actually help to prevent the severe cytokine release syndrome. And then we do the follow-up as indicated. What I can tell you and report, and we'll be reporting this at least first cohort of patients, cohort 1 at the ASH meeting this December, is that the pharmacokinetics, the engraftment of T cells that we see in this setting, looks comparable to what we are seeing in patients with CTL019. And just to show you, one patient that we've disclosed, which is a patient who had a fairly aggressive disease. He's a 66-year-old male who had 11 prior lines of therapy, progressing on his last therapy, had bone marrow with 70% involvement. You can see the genetics there. And this patient received basically 2 × 10⁸ CAR cells, no lymphodepleting chemotherapy. And he developed a grade 3 CRS that responded quite nicely to tocilizumab. And you can see on the right a robust engraftment of his CAR T cells, reaching as high as 95% of his peripheral blood T cells at the peak of the response. You can see on the lower left, that those cells are CAR positive. This is at day 7, quite robust expansion. Same thing shown by the qPCR, quantitative PCR results, and including parallel in the bone marrow. We saw quite high levels of T cell engraftment. And this patient went on to really, show flow cytometry. This is showing his day 28 bone marrow, where the malignant clone that's shown in that upper left, gate really is, undetectable in this flow. This patient has gone on, and actually I should say is actually beyond VGPR, is now in a stringent CR. I think I forgot to update this slide. But basically, you can see here the kinetics really of his M spike declining, which is probably representative more or less of his antibody clearance and the reductions in his free light chain as well as urine M spike. So just in the final few minutes, how do we expand this beyond heme malignancies? So as I mentioned, lots of different efforts. Everybody's working on CD19. More and more people are working on BCMA. What about solid tumors? Lots of antigens being developed, including at our institution at Penn. And I think one of the big challenges in solid tumors is, as I alluded to from the beginning, is really target selection. Target is important. And so we are, very cognizant of these early studies with CARs, including the top one here in 2006, which was a first-generation no co-stimulation CAR targeting carbonic anhydrase 9 that was thought to be restricted to renal cell carcinoma, but these patients went on to develop evidence of biliary sort of targeting due to a very low level expression of this. And then this has been observed with HER2 in terms of ERB2, and then even in targeting things like CEA, in colorectal cancer. So target restriction is important. So we've been looking at mesothelin CARTs. I'm going to just show you, we've moved this forward. This is all published, but essentially showing again that we can target this. This is with hopefully everybody can hear me. Oh, sorry, that's not directed to me. So the just showing that we can in fact make a co-stimulated CAR that works. The challenge with this is that mesothelin is expressed by normal tissues, and we knew that going in. It's expressed on normal mesothelial cells that line the pleura, the pericardium, and the peritoneum. But it is expressed on a range of different cancers, including ovarian, pancreatic, and mesothelioma. So we've actually done a phase 1 clinical trial using these lentivirally engineered cells. We also did a non-stable transfection-based approach initially with this. And this is just showing the sort of interim results for this mesothelin trial. But you can see, unlike what I showed you for the CD19 CAR, the levels of engraftment here in the blood, these are, again, vector copies per microgram of blood, are orders of magnitude lower engraftment than what we see in the CD19 or BCMA setting. And even with cytoxin for lymphodepletion, it certainly enhances the level of engraftment to some extent, but still nowhere near the level of engraftment. We haven't seen much in the way of effect to partial responses in our initial cohort of 6 patients. And we have basically, considered some possibilities. We haven't seen long-term persistence. We are using a murine single-chain FV for this, and so one of the efforts is to try to switch this to a human single-chain FV, because perhaps immunogenicity, which has been shown to be important in some CARs, could be playing a role. But I think the other challenge is that in solid tumors, and this is work that we've been collaborating with Steve Albelda here at the University of Pennsylvania, who's working in mesothelioma, that in this preclinical model of mesothelioma, you can see that when you put these T cells into this model, while they can have a modest effect on the tumor, the T cells ultimately become hypofunctional and cease functioning over time. And so this loss of function is something that I think we need to spend a lot more effort into understanding. Some of it may be related to immune checkpoints and regulation like that, but I don't think we really understand these mechanisms at all. And then I think finally to end, I just want to talk about sort of the future, how you actually bring this to patients, right? Because one thing to do this in a academic center like Penn, so we treated our first patients with this in July of 2010. And the question is, we've only treated maybe a few hundred patients at this point. But how do you treat thousands of patients a year with this type of very complex therapy? And I think, this is one of the challenges that, hopefully over the next few years we will figure out whether we can do this. But I think the important thing that we recognize is that, really the existing sort of biotech, pharma, medical device industry really has no experience with this unique type of therapeutic approach. And that really it may be that you need a separate sort of area of global healthcare really around cellular therapeutics. They really are very, very different. And in particular, some of the issues to think about is this is a very patient-specific model unless universal sort of allogeneic CARs can be made to work. And so every product is a unique product. And is centralized manufacturing versus blood bank sort of distributed manufacturing the way to do this? And I don't think we know the answer to that yet. The biotech sort of industry is really focused around a centralized manufacturing approach currently. We'll see if that's successful. But I think we've certainly demonstrated that outscaling and distributed manufacturing can be done. And the first cell therapy, basically T-cell therapy, if you think about it, is bone marrow transplantation, and we've been able to do millions. So there is a way to deliver this, if it can be successful. The other challenge is it's quite expensive, and complex to manufacture these. And I think we can learn, and this is an area that I think that's going to be very heavily, explored in the future, is can we learn from, right now we're making, I kind of use the Lamborghini analogy. It's a very highly customized, handmade product right now. And can we really switch this to a Ford model where we can actually do automated manufacturing that will probably be not only more efficient, but also perhaps even more consistent. And so with that, I'll just have to acknowledge the many, many people who have been involved in these studies. It takes a village to do this, particularly Carl June and David Porter, Steve Grupp, Noel Fry. These are the clinical investigators who really have pushed forward with these studies, and Adam Cohen in our BCMA trial, and then Bruce Levine, who's really pioneered a lot of the manufacturing. I have to thank the patients, who are really brave enough to volunteer for these first-in-human studies, both adult and children. And with that, I guess I'll do the questions.
Okay, thank you so much, Doctor. Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit the questions by typing them in the Q&A box, which is located in the bottom left-hand corner of your screen. We'll try to answer as many questions as possible. And let's see, we're still, so we'll wait a moment to see if anyone has a question coming in. But Doctor, do you have any final comments in lieu of any questions that you'd just like to get out?
Any final comments? I think I commented enough. Happily answer questions.
Okay, well, as there looks like there are no more questions for today, I'd once again like to thank you, Dr. Milone, for your presentation. Today's webcast will be available for on-demand viewing through February 2017. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward this announcement to your colleagues who may have missed today's event. See you next time. Once again, this is Christina from LabRoots. Have a great day.